So we have ended our lecture. You should be able to see my screen now. We have ended our lecture on that slide last time. It was quite interesting slide to read, that how biology is evolving in one direction. The rules, there are many rules out there. And then the information is generated by these people. And then the basics of DNA we came to know. And then everything is moving for one thing, and that one thing is genome editing. The ultimate purpose of knowing natural phenomena, the ultimate agenda of discovering a natural phenomena is to edit that phenomena, is to change that phenomena for our own sake. I remember a dialogue from one of the movies where that we control the time. Oh, Indian movie थी, ऋतिक रोशन की, जिसमें वो सुपर हीरो बनता है. क्या नाम था उसका, जो भी नाम था, क्रिश. We control the time. For what purpose? The villain was saying like this. हम वक्त को इसलिए मैंने ये टाइम मशीन बनाई है ताकि हम उसे कंट्रोल कर सकें, हम उसको चेंज कर सकें. So we are trying to understand the coding of life so that we can manipulate it. And we have evolved and came to know about a wonderful phenomena called by the CRISPR. I want you people to go through the abstracts of these things. I don't want you to, like, it's your own choice. If you are feeling comfortable, you can go in detail of these things. But if an area of research is of your interest, if you can say that this is my area of research, then you should know everything about that stuff. इसमें कोई दो राय नहीं है. There is no easier way, there is no shortcut in that. So if you believe, if you sense that genome editing or CRISPR Cas9 system and learning about these things is your area, I can show you the guidelines, the big headings, and then it is your job to thoroughly investigate each of all those aspects in detail if you want to stay in that field for the rest of your life. You want to take this as a career, you are supposed to know everything about that stuff as a student. So it appears in 1987 when the clusters, repetitive sequences were discovered. And remember, it was discovered in bacteria. There was no mammal, no concept of mammalian system out there. In 2000, people reported that it is present in every prokaryote. So most of the prokaryote shows these clustered repetitive sequences in their cells. In 2005, people came to know that this clustered repetitive sequences are actually involved in a fight against the invaders, particularly against viruses. So it is like a viral, it is like an antiviral defense mechanism evolved by the prokaryotes. So when a virus entered into a bacterial system, the bacteria has a mean. The bacteria try to understand the virus. And you already know all these things, that the virus actually send the genetic material to the bacteria. So the protein codes stay outside, and it's only the genetic material which enters into the cytoplasm. So the bacterial genome needs to find an answer against this. So this is the genome of bacteria. This is the genome of the virus. So genome of bacteria versus DNA or RNA of the virus. So the CRISPR system is a tool which was evolved by prokaryotic system. They understand, they try to learn and read the genetical sequence of the virus and then create repetitive sequences coming from different origins of the genome of bacteria, put them together based on the nature of virus. And those repetitive sequences then able to bind with a particular virus. If the virus is changed, the bacteria create another combination of those repetitive sequences. And that new combination of repetitive sequences finds a match with the new virus. So no matter a virus A is coming, a virus B is coming, a virus C is coming, or a virus with a newer version of RNA, a virus with a newer version of DNA, most of the time, the bacteria or prokaryotic system able to generate clustered repetitive sequences which able to successfully match with the sequence of virus, and it able to destroy the virus. So the first thing you need to know, everything related to CRISPR is not naturally present in the mammalian system, full stop. So it's not present naturally in the mammalians. We are the mammals and the higher organisms and humans, we have another ways of dealing with the viruses. We don't have CRISPR system in our cells. So everything was there in prokaryotes. And let me add a couple of more papers. So first reported the repeats, then found it is present in prokaryotes like a universal immune system of bacteria against viruses, means against invading genetic material, against invading RNA or against invading DNA. It's an immune function. And then in 2010, it was proposed that if we associate a Cas9 protein, remember the FOC1 system. The FOC1 is a nuclease. It's an unguided missile. And you have added the DNA binding domain to send the FOC1 at the place of your own choice. Similarly, Cas9 is a nuclease. It's a big protein. It's a nuclease present in bacteria, not in humans, not in mammals, only in bacteria. Whatever we are talking, these things are present in prokaryotic system. So in 2010, these people able to target, and they successfully create a DSP, a double-stranded break in the genome, I believe, of bacteria with the help of a Cas9 supplemented with a guide RNA. The guide RNA is the sequence of RNA which was created by the prokaryotic system to target the virus. So remember, there are two types of DNA out there. One is the viral DNA, the other is the bacterial DNA. The bacterial DNA creates the guide RNA which can find a match with the target viral DNA. And if that bacterial guide RNA sequence is supplemented with a Cas9 protein, that system will effectively cuts or create a double-stranded DNA break on the viral DNA. So the viral DNA is gone. After that, it will not be able to, do the function of the virus. In 2011, this is a groundbreaking work somehow. Some people, these people, heterologous expression of CRISPR type 2 system. Heterologous expression means that something is not produced in a cell naturally. As we know, the CRISPR system is not naturally produced by our genome, by humans and by mammals. So we use bacterial genes, or we used the information we collected from bacteria, and then we try to produce CRISPR Cas9 or Cas9 proteins right within the mammalian cell. So if we install those genes into humans, the human cells will start producing the Cas9 protein. Now the Cas9 protein produced in human cells will be considered as heterologous because we are not producing it naturally. There is no gene in humans which can produce Cas9. It only belongs to the prokaryotic system. And after that, these brilliant people took that stuff to the skies. 2012, it was proposed that the CRISPR system can be used to edit the genome. And then this man, most of the papers, the founding papers about the technique came from this man. You will always see the Zhang and Zhang and Zhang and every the Zhang. He managed successfully edit the genome of a eukaryotic cell. Remember, the eukaryotic cells are not supposed to produce CRISPR as well as the Cas9 protein. So they use bacterial information to create CRISPR Cas9 system within the eukaryotic cells, and then successfully able to edit the genome of a eukaryotic cell. You can see the organism they have used. You can see which type of cells they have used. This is your job to go and see the paper. And after that, the structure was also reported by that group. That what was the crystal structure of the Cas9 and guide RNA system. The gRNA means the guide RNA. I will show you ye kya hota hai. One extra thing you need to know here, it's a little bit, maybe it might be very easy for you, you already know that thing. But just some basic molecular biology stuff you should know. If I know a gene from bacteria which can encode Cas9, using that sequence or after amplifying that sequence from bacteria, if I install that copy into human genome, if we send that plasmid into human cells, it will not going to produce the Cas9 protein. It will not going to express in an efficient way into human cells. I wish this should be a class so I able to get your answers and your opinion on that thing. The reason why, because I just have to tell you the answers right away. I'm sorry. The bacterial gene installed or sent to the human systems, the bacterial gene sent to the human systems will not going to be translated efficiently. And the difference is because of the difference in the ability of the two systems to use the codons. I wish you should be able to understand that. You should try to understand that concept completely. मैं इसको मज़ीद बोलता हूँ इसके ऊपर. When bacterial gene produce messenger RNA, that messenger RNA contains the codons, and those codons will be utilized by the translational machinery to produce protein, full stop. The humans produce messenger RNA. It is captured by the ribosomes and utilize the information present on the messenger RNA to produce proteins using the codons present on the messenger RNA. The amino acids are universal. They are same in human cells as well as in the bacterial cells. So both the cells have the same amino acids. But the codons and the choice of codons are different in bacteria and in humans. One amino acid can be coded by multiple codons. This is very basic information. One amino acid can be coded by multiple codons. Let me write these things to help you understand this very important concept. This is the amino acid called A. It can be coded by a codon called X, Y, and Z. So the codon X will incorporate amino acid A. The codon Y can also incorporate the amino acid A during the translation. The codon Z will also incorporate the amino acid A. You know that one codon is made up of 3 base pairs. I'm just using the symbols. I believe that the rest of the things you understand well. Now, one amino acid can be incorporated, can be installed through 3 different codons. When we are in bacteria, the bacteria or bacterial translation system show preference to one of the 3 codons for the translation. The bacteria prefers the X to code amino acid called A. This is called Codon Preferences. Bacteria prefer to use, whenever bacteria wants to install amino acid A into the protein, bacteria use the codon called X. Bacteria do not like to use Y or Z. This is the preference of bacteria. In humans, for example, in humans utilize or prefer, whenever the humans wants to encode, wants to incorporate the amino acid A, because the amino acids are constant, they are universal. Humans prefer to use the codon called Y. So when you extracted a gene from bacteria, every A in the protein will be supplied by the codon called X. Every A of the protein you want to create is associated with a codon called X. When you put that stuff to the human system, when you put that plasmid to the human will not be able to prefer the codon called X. So that is why it will not be able to incorporate the amino acid A. If we want that human system should be able to produce the bacterial protein comfortably, we should replace the X, every X, with the Y. So when that bacterial gene enters into humans, every X codon will be replaced by the Y, and that is why human system will be able to install amino acid called A. So जो भी आप बैक्टीरिया से जीन एक्सट्रैक्ट करेंगे, that gene is optimized to be used in the translation machinery by bacteria. If you want to use the same gene in human system, you have to change all the codons in such a way. This is called codon optimization. You need to change the codons in such a way so the preferences will be shifted to human side. So the humans can use the same gene to produce the same protein. So in total, it's a very important question. Somebody will ask you in the upcoming career options. Same protein produced by the bacteria and by the humans. 100% same protein produced by the bacteria, bacterial translation system, produced by the human translation system, will have a different sequence of DNA. If you are getting this line, I will be very happy. I'm going to repeat it once again. Same protein produced by bacteria and humans. So humans and bacteria are producing a Cas9 protein. For example, bacteria is producing a Cas9 protein, human cells are producing Cas9 protein. The genes, the protein is same. The genes which are producing the proteins have different sequence in bacteria as compared to the sequence in humans. Because in humans, if we want to produce the same protein, we have to change the codons to prefer in such a way so human translation system can use it efficiently. Okay, please read that thing further or ask me more about that concept. This is very important. I believe it should be taught in the other things, other ways, but or the classes, but if not, then please do ask that thing to me. Further, how the CRISPR Cas9 system actually looks. What are the important ingredients and components? Okay, I will be taking a 2 minutes break. Okay, I will be sending you the password of the next session through WhatsApp, so you will connect on the other side after the 2 minutes, okay, using that information, and then I will complete that lecture. In the meanwhile, keep pondering of that stuff which I just told you. I have recorded that lecture, so please go that stuff again and again. You need to be very clear. Want to express the same gene in bacteria? You want to express the same protein in bacteria? You want to express the same protein in fungus? You want to express the same protein in humans? These three sequences will be different because bacteria uses a different preferences for a same amino acid. Fungus use a different preferences. Fungus has its own preferences to install the same amino acid. Humans have their own preferences to install those. The trouble is because one amino acid can be incorporated by multiple codons. So which of the codon will be used to install one amino acid? It depends on the species, on the system. Okay, see you on the other side in the second session. Okay, I'm stopping this one. I'm stopping my screen and recording. Okay, bye bye. See you after a short break. इससे पहले कि I go for the next slide. If anybody have any question, just feel free and let me know. यहाँ तक story समझ में आ रही है सारी? If you are getting the things nice and fine, then I will go to the main slide of that topic to show you the different components present in the CRISPR Cas9 system. So I believe everybody is here. So somebody have the question, I guess. So does the codon preference change or it remains constant? Codon preference के बारे में पहली बात याद रखें. The first thing you need to remember about the codon preference, that it's a preference. So it is like if there is, I told you and I show you, there is an amino acid called A. It can be coded by 3 amino acids. It can be coded by 3 codons. So the codon 1 has like 100% preference. The codon 2 is like 60% preference. The codon 3 is like 30% preference. So bacterial system will be comfortable if the codon 1 is present out there. The translation machinery will not be inefficient. That thing stays constant. ये constant रहता है. The same 3 amino acids. Let's go in the human system. The third one has a 100% preference. The second one has a 60% preference. The first one has a like 20% preference. So the first one is working nice and fine in bacteria, but when it is present in the human system, the human translation machinery will find it difficult to incorporate the amino acid A in the protein. The efficiency will be less, and ultimately the expression will not be good enough to do the job. So that preference is evolved over the period of like a lot of time, and that stays constant for different species. So bacteria, another question. So bacterial system cannot be used to synthesize human Cas9 protein. Bacterial genes, the sequence of the Cas9 protein, ये बात important, the sequence of the Cas9 protein which can produce or which can express in bacteria is specific to bacteria, and it is different from the sequence of the Cas9 gene which can produce the same protein in humans. So in humans, the Cas9 protein will be produced by a different sequence. In bacteria, the same Cas9 protein will be produced by a different sequence. If you want to produce a human protein in bacteria, you need to remember first that the bacterial translation machinery is less evolved as compared to the eukaryotic system. The folding of proteins, the architecture of protein will not be built well if it is far too complicated. And to get everything clear, the human insulin is produced in bacteria successfully, and bacteria have no trouble in producing human insulin. You know why? Because insulin is very simple protein. Bacteria don't need to, fold it in a complicated ways. But that is important, which I'm going to tell you. But the sequence of the DNA which is used to produce insulin in bacteria, that sequence is different, and it is optimized to work in bacterial system. That sequence is not a match with the sequence of DNA we are carrying. Because whatever the sequence of DNA we are carrying is optimized to produce the same insulin with the different codons based on our own preference. When we want to install, if the human insulin gene is installed in bacteria, the bacterial translation system will not be able to use that gene because the preference will not support the bacterial system. So that is why we can produce human proteins within bacteria, but we need to change the preference accordingly. Like if we want to produce a bacterial protein in human system, we have to change the preferences. We have to change the codons accordingly. So the human translation machinery can easily express that protein in humans. Farhan ने question किया है, will the human cell environment affect how the Cas9 protein is folded or other? Of course, the human capacity, the human cell's translational ability and the post-translational modification affects a lot. And the people who are working on the expression of the proteins in humans or heterologous expression, their job is to know all these complications and then fix them. ये पता कैसे लगा होगा? At the start, लोगों ने बैक्टीरिया का DNA एम्पलीफाई किया, humans में इंस्टॉल कर दिया, mammalian cells में इंस्टॉल कर दिया. But they were expecting to produce the bacterial protein, but they failed to produce it efficiently. they might be able to get a very little bit of expression of that protein, which they later came to know about through the concept of codon optimization. When they optimize the DNA sequence according to humans, according to mammalian system, they're able to get the expression in a wonderful way. And human system do affect, it's very important. This is the job of the people who are studying the heterologous expression. For example, if you want to produce a bacterial protein in humans, you just install the bacterial gene into a plasmid and send it to the human or mammalian cell. If you get 70 to 80% efficiency of a translation, you don't need to worry at all. You can proceed with the same thing. But if you are not getting the expression, the first you need to optimize the codons for human system. Let's suppose you optimize the codons and you still not be able to get an expression in a better way. You need to see the other things, and those are the things which Farhan is asking. The new environment, the complicated one, and the possible post-translation modifications. So all these things are needed to be addressed in order to express a system. Okay, so let me take you to the slide which explain the different components of the system. The green stuff, all that green portion, this is the Cas9 protein. Let me tell you, the scale of the picture is not correct. The Cas9 protein is very big. Just go and see the size of that protein. It's pretty big, and DNA is very, very small as compared to that protein. But just for the sake of a cartoon, just for the sake of a picture, they showed this thing in this way. So this is the Cas9 protein, and that protein actually moves, is capable, it's a DNA binding protein without any specific DNA binding domain. So it can bind itself to the DNA, but it slides onto the DNA. And you know, DNA is not like a railway track. Railway track hota na, those strands asa jari. It's like twisted around. So the Cas9 protein, you can imagine, is actually moving like this around the DNA. It's moving like a twisted form on the DNA strand. It does not slow down, it just kept on moving. Because the Cas9 protein do not know where to stop. It has no DNA binding capacity, but it can stay on the DNA. It can slide. जैसे रोलर कोस्टर होती है ना, ऐसे 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 DNA के ऊपर ऐसे move करता रहता. The thing which can give this Cas9 protein the ability to sense and to stop at that one place is called as guide RNA. So the second important component is called as the guide RNA. Guide RNA has a like loop structure, and you know how the loop structure is created. The loop structure is created because of the presence of the certain specific sequences here. For example, the A is here, so that means the T is present as well on the same strand. The C is here, that means a G is present on the same strand. So that will create a self-complementation and creates a loop. That loops allow the guide RNA to be sit comfortably into the pocket created for that purpose. The Cas9 do carry a pocket which can be effectively filled by this structure, this loop structure of the guide RNA. So this part of the guide RNA is responsible to attach itself with the Cas9 system. The other part of the guide RNA, this part of the guide RNA, the blue one, is responsible to find a complementation, a matching sequence with a certain genome. So one portion of the guide RNA allows the guide RNA to fit into or to load onto the Cas9 system. The other portion allows the guide RNA to find a match with a genome sequence. This system traditionally works that the matching sequence should start with a PAM sequence. The matching sequence should start with this yellow one called the PAM sequence. PAM sequence is called like NGG. Anything, N means any base pair, and you know the G and then the G. This is like a password. This is like a key for the Cas9 system to slow down and to sense that this might be a place where I should see, where I should look at the matching sequence. It's like a password. Once everything is there, the Cas9 system, when it is rolling over on the genomic DNA, it slows down here. The Cas9 protein pocket create a comparatively higher temperature because its movement and vibrations. You know, proteins used to vibrate, like move like this. So this vibration, and there is a DNA present in that pocket, will create vibrations. Means high kinetic energy. Kinetic energy का मतलब है ज़्यादा temperature. The average kinetic energy of the system increases, and that create high temperature right inside that pocket or domain of the Cas9 system, allows the DNA melting. And that melting allows the annealing of the guide RNA with the system. Once that stuff is in place, the nuclease activity of the Cas9 is is activated. Cas9 is the nuclease, so it can create a double-stranded DNA break. And rest of the story, you know. If you do not interfere, what will happen? Non-homologous end joining. The natural DNA repair mechanism will try to fix, will try to correct that double-stranded break, leaving the mutation of some base pairs. It will add or it will remove some base pairs. Together it is called as indel. Indel, insertions and deletions. There might be insertions, there might be deletions. So the mutation will be there. But if you interfere that system and supply a donor DNA, like the blue one here, so that donor DNA will be incorporated into the double-stranded DNA break. And you will be able to edit the genome at the place of your own choice. Let's see the same picture in a different way. Not the different way, I mean, just in the same way. The correct dynamics of that machinery, the presence of the guide RNA, the annealing of the guide RNA to the target sequence creates a double-stranded DNA break here. Actually, Cas9 protein has two different domains. This is important. We will study that CRISPR Cas9 system more after in the next lecture. The one side of the Cas9 creates the break on one side of the strand. The one side of the Cas9 cuts one side of the DNA. The other side cuts another side. Creation of a break on a single strand is called as nick. It is called as nick. When two nicks are there on the opposite strand, you can guess it will be a double-stranded break. If you provide a donor DNA at that time, it will technically be incorporated with a much higher efficiency as compared to the zinc finger, as compared to the talons. Because the talons and zinc finger required extreme cloning. It just required what? Just required the guide RNA and the Cas9 system over there. It will going to edit any target place, but still the efficiency is not very good. I mean, it's not something which you can proud of, but it's way much easier, less laborious and more efficient as compared to the other two systems. People are not looking to zinc fingers and talons anymore. They're done. With that system, until today, people have successfully added bacteria, fungus, and mammals and plants. It's used in all, and of course you can guess. In plants, the system promoters might be different. In mammals, they are different. In bacteria and fungus, they might be different. This is the technical expertise which should be there after the undergrad student. The undergrad student should get a sense that when I will be using that technique in mammals, I need to optimize my sequences. I need to optimize my options, my promoters, and my vectors in a different way. If I will be ended up in plant sciences and I will be using the CRISPR Cas9 system, I need to adopt that system in a plant's way, which can work in plant in a better way. This is the speciality which should be there in the future. We heard the rumors a few years ago that the Chinese scientists had genetically modified human embryo. They're trying to use it with the help of using CRISPR Cas9, but there was no authentic paper out there because it cannot be published. You can guess it's unethical. So it will not be published anywhere. So somebody can do that thing privately. Try to edit the human embryo at the single cell stage using the CRISPR Cas9 system. And you can get, you know why I told you in the earlier slide that this is the ultimate objective of developing that technique, to edit the genome of human in a way we want. So everybody would like to have that thing. If it is not allowed ethically today, people might be doing it without knowledge of anybody else. But they have tried to explain that actually they are doing, I will explain. They are saying that we are trying to fix a very bad disease called thalassemia to make it more ethically acceptable. It's a potentially fatal disease, blood disorder. We are using unviable human embryos. The human embryos which were left over after IVF and other options. And they said that we are trying to replace the mutated HBV gene with the corrected version with that stuff. We're trying to replace, we're targeting one particular bad thing in the human genome, where in the embryos, the leftover embryos. And if we successfully added the one stuff, we can implant that cell into the womb of a surrogate mother to develop a baby without any fear of thalassemia. That looked very fancy, it looked very attractive, a solution. But when you are trying that stuff, there is a term which is called off-site activity. Off-site activity means even if you have like 20% or 30% success rate of targeting a cell, 30% success. What about the 70% inefficient targeting or off-site activity? What if the guide RNA is ended somewhere else with a similar or closely similar sequences? What if it is destroying some important other components, some important other places in the genome of the baby? So you have fixed the thalassemia, but since you can't control the off-site activity and the efficiency today, so you might be having a lot of unwanted troubles in the genome, which you don't know. So we can't risk to proceed with the development of such zygotes. Until unless, when we can say we can go further, when we know The efficiency rate is like 99.9% or something like that. And we know where we are sending and we know what we are doing. As long as the efficiency level is not that high, the chance of having a baby with unwanted troubles will always be there. It stays unethical. But in plants, you can experiment anything. For example, this thing is easily approved by regulations. There are a lot of regulations in genetic modified organisms in USA. The mushrooms, the color is changed because of the polyphenol oxidase. An enzyme present in the mushroom changed the color and it changed the storage capacity. So ये खराब हो जाता है बड़ी जल्दी because of that enzyme. This enzyme has 6 components. So the scientists created a CRISPR Cas9 targeting system to destroy one of the exon of this PPO gene. So it reduces the activity 30%. So now the people can store the same mushroom like for a longer period of time. Similar things can be done. And more things we will study in the next class, that how people have evolved the CRISPR Cas9 system nowadays. Okay, so that's it from my side today. I advise you people that you should go into the details of these papers which I am mentioning here, just to feel that how these people are working and about the details. For example, when I said you that it is done in the bacteria, you should try a couple of papers where CRISPR Cas9 system is used to edit the bacterial genome for good purpose, for bad purpose, for producing X, producing Y in fungus and in mammals and in plants. And you can guess the genetically modified organisms, GMO foods. The people might be creating GMO food using some CRISPR Cas9 system. You should be able to fill all those blanks in your private folders and in your intellectual library. I don't want you to remember those papers, but at least you should try a couple of papers of each of these things. Okay, so that's it from my side. If you have any question, you can ask that question to me right now. I am stopping my screen, but I am ready to take your questions. You can speak as well. If you don't like, you can write as well. Ji Hamza.
कि CRISPR Cas9 सिस्टम के अंदर Cas9 प्रोटीन के अंदर हम गाइड आरएनए किस तरह डालते हैं, व्हाट डिटरमाइंस कि गाइड आरएनए कौन सा वाला प्रोटीन?
Okay, Shabash. There are many ways we are doing this thing nowadays, but let me tell you the most initial ways. We are creating a vector that carries a gene which can encode Cas9 protein. Okay, so one part of the vector is producing a Cas9 protein. It is the same thing and the same procedure through which we produce or we express any protein in bacterial system, जो आप पढ़ते आ रहे हैं. Hamza, are you here? Hamza, can you hear me? So you can see the recorded answer, by the way. So sorry, Hamza, let me answer you once again. We create a vector. One part of the vector carries the Cas9 gene under the promoter, and that promoter is the one which can express and push the transcription in the bacterial system. The Cas9 messenger RNA, and then it's again same traditional way of producing a protein in a bacteria. The same vector contains another site, another cloning site or another site, which is under the control of a different promoter. If the promoter recruits RNA pol 2, it will produce a messenger RNA. Okay? If a promoter recruits RNA pol 2, तो उसके आगे क्या होना चाहिए? Protein coding gene. If a promoter recruits RNA pol 3, if a promoter recruits RNA pol 3, then what should be the next thing? RNA. Okay, RNA pol 3 is famous control for the transcription of RNA. Non-coding RNA, other type of RNA. So the same vector, one side is designed in a traditional way to produce Cas9, and the other side is created under the control of pol 3, RNA pol 3. वो वाले promoter अलग होते हैं, like U6 promoter. I'm just giving you one example. But those are the promoters which recruit not the RNA pol 2 but the RNA pol 3. So whatever the sequence after that will be transcribed into an RNA, and that will be a guide RNA. So one vector is producing the two things in the system. They will connect and they make a functional CRISPR Cas9 system. The other way is a little bit more, it's more smart. People make a stable cell line, sending CRISPR Cas9 into the genome of the mammalian cell. So the mammalian cell genome contains the CRISPR Cas9 gene. Every cell. You don't need to send. All you need to do is to send a vector with a RNA pol 3 based expression vector, which will go and create the guide RNA. So you can send an X guide RNA to knock out the X gene. You can send the Y guide RNA to knock out the Y gene. You can send the Z guide RNA because the Cas9 system is there already. This is another way. Okay. So people are working on finding an efficient way of sending these things into the cell. You know why? To increase the efficiency. So we can say that every time we send a stuff, it will not only enter into the cells, but it will able to successfully edit the genome. Remember, editing the genome is the last thing. There's a lot of stuff we need to do at the start. Everything should be perfect. That will reduce the efficiency over the period of time. If you are less efficient in sending the ingredients within the cell, mammalian cell पे काम कर रहे, you have to send something across the cell membrane, है ना? Cell membrane is not easy to cross thing. If you are inefficient at that stage, you can guess what will happen after that. उसके बाद क्या होना है? उसके बाद उसने साइटोप्लाज्म में सर्वाइव करके डबल न्यूक्लियर मेम्ब्रेन क्रॉस करनी है, and then able to deliver the material over there. So if you are not able to be efficient in the first state, you will not be able to get a higher efficiency. So whatever the things left in the nucleus is nothing. So people are working in different aspects. First, wonderful delivery system. So you can send more and more things within the cells, controlling the things well to increase the efficiency. Okay. So thank you very much. अभी हमारे पास कोई क्वेश्चन नहीं है और anybody wants to speak? आप लोग बोलते नहीं है यार, मैं आप लोगों को मिस करता रहता हूँ, बहुत शर्मीले। जी जैनब, शाबाश।
सर एक दफा प्लीज, आपने दो मेथड्स बताए हैं। एक तो आपने कहा था ना कि एक वेक्टर हम क्रिएट करें जिसमें Cas9 की जीन और दूसरे प्रमोटर को की एबिलिटी है आरएनए पॉलीमरेज 3 को रिक्रूट करने की, और एक और मेथड आपने बताया था वो मुझे समझ नहीं आया। और पहले मैंने आपसे पूछना था कि आप कह रहे थे जिस वक्त Cas9 का कॉम्प्लेक्स और गाइड आरएनए का कॉम्प्लेक्स स्टॉप करता है पर्टिकुलर पॉइंट ऑफ द डीएनए, तो उस वक्त जो टेम्परेचर राइज है वो टेम्परेचर राइज से मेल्टिंग होती है डीएनए की, और फिर वो अनील करता है गाइड आरएनए के साथ, और उस गाइड आरएनए की अनीलिंग डबल स्टैंडर्ड ब्रेक क्रिएट करती है। या आप ये सजेस्ट कर रहे थे ना कि वो अनीलिंग डबल स्टैंडर्ड ब्रेक क्रिएट करती है और फिर आपने जो भी सीक्वेंस भेजा होगा वो इंस्टॉल हो सकता है जीनोम के अंदर।
जी ठीक है। एक्चुअली वो द Cas9 सिस्टम की जो वर्किंग कॉन्फर्मेशन है जी, Cas9 सिस्टम में क्या चीज होती है? Cas9 सिस्टम इज अ न्यूक्लियस जी, it is supposed to cut the double-stranded DNA, but it will cut the double-stranded DNA when it will become like this. For example, don't, there's a funny face, it become like, you have to turn and twist the Cas9 system to that state. Once it is here, it will destroy, it will create a double-stranded DNA break. So the presence of the guide RNA loop, the presence of the match will actually turn and twist the Cas9 system in a working conformation to induce a double-stranded DNA break. It's more easier way to understand the thing. So all these things should be in place for one purpose, and the purpose is to create a working conformation for the Cas9. Okay, and there are two ways. The first way, if you remember, the good enough, the plasmid, you should browse, or I should show you in the next class to explain you about the vectors which are used. Okay, then how different components of vectors are there. So the first thing you understand. The second thing you don't understand is that if I send a Cas9 gene into a human cell, into the genome of the human cell, अगर मैं Cas9 gene किसी human genome में integrate कर दूं, तो human cells के पास Cas9 protein होएगा ना already, because it is already part of the cells. अब मैं ये वाले cell बेच सकता हूँ लोगों को. Now I can sell that cell to the people. बेच सकता हूँ ना मैं कि आपने कोई जीन डिलीट करना है, कोई जीन एडिट करना है, तो हमारे वाला सेल यूज कर लें. आपको इसमें Cas9 भेजने की जरूरत नहीं है, ये ऑलरेडी जीनोम के अंदर है. All you need to do is a vector which can produce only the guide RNA. अब ये बात समझने वाली, जो हमजा ने सवाल पूछा था, गाइड RNA प्रोड्यूस करना और बात है. It is produced under a different promoter, और प्रोटीन यानी कि Cas9 मैसेंजर RNA प्रोड्यूस करने के लिए हमें RNA pol 2 का प्रोमोटर चाहिए. ये आपने पढ़े हुए सारे RDT में पढ़े होंगे, मेरा ख्याल है आपको आते होंगे वेक्टर्स, प्रोमोटर्स सारे. वरना मैं अगले लेक्चर में दिखा दूंगा आपको. ठीक है, हम ब्राउज करके इंटरनेट से देख लेंगे, नो प्रॉब्लम. शाबाश. कहाँ है मेरी इतनी प्यारी क्लास, सारी की सारी चुप चुप रहते हैं, पता नहीं सो तो नहीं रहे होते क्लास में सारे. एक दिन सब लोग माइक जला के फिर बातें करेंगे. ओके, if I'm not having any more question, but I will always try my best to answer your questions on and off. But if I don't able to answer your questions immediately, just hold your patience. I'll try my best. Okay, so you can ask your question in the next class on Friday as well. Just try to keep that information and just try to build. I just want your concepts to be very perfect. So whatever the knowledge you will accumulate, it should create a perfectly, stable CRISPR Cas9 understanding in your mind. तो ये basics बिल्कुल clarity के साथ. अभी मैंने आपको ये नहीं बताया, it's not understand that the best way to use me. मैंने आपको ये नहीं बताया कि जब माउस में उन्होंने किया होगा तो कौन से वेक्टर्स इस्तेमाल किए होंगे, कौन सी स्ट्रेटेजी इस्तेमाल. दिस इज समथिंग व्हिच यू नीड टू एक्सप्लोर, टू शेयर विद मी एंड टू शेयर विद माय अदर फ्रेंड्स एंड योर कोलीग्स एज वेल. ओके, लिटी गिटी से होती हैं छोटी मोटी. Okay, so since we don't have any other questions, so I wish all of you a very good and a safe time out there. Please take a good care of yourself, and I miss you